Hello everyone and hello from Malaysia. Welcome to our ABSA Biotechnology Student Association Webinar Series. Thank you very much for your participation today. You have joined our webinar on cells in modern medicine. We will begin our program shortly. I would like to kick off today's program by reminding you of some of the housekeeping items as seen on the screen. First of all, please turn off your microphone during the session and only unmute when required to speak. Please turn on your camera so that we can see each other. If you have any questions or comments, please feel free to message us through the chat box. We will try our best to address your questions. The chat box is also there for you to introduce yourself. Please give us your name and your organization. Last but not least, no phone calls or distraction during the session. Note that this session will be recorded and live streaming is done through YouTube. This recording will be saved on our YouTube channel right after the session. An evaluation survey will appear at the end of our session. Please spend some time to provide your feedback, as it will be very meaningful for us to improve our next session. Thank you very much for your attention, and once again, welcome all to our webinar organized by ABSA Biotechnology Student Association. My name is Dan Aikam. I am proud to tell you that I am a BSc Biotechnology degree program student at ABSA University. It is a great honor for me to serve as the MC for the webinar. If anyone has any issue with audio or video, please use the chat box and we will do our best to help you. To start the session, I would like to invite Ms. Vivian, the ABSA Vice President, to deliver her welcoming remarks.
Thank you, MC, Mr. Tan Aikam. Hello, everyone, and greetings from Malaysia. Our honorable speaker of the day, Associate Professor Dr. Rajesh Ramasamy, Head of Department of Pathology, University Putra Malaysia, UPM. Senior Professor M. Ravichandran, Dean of Faculty of Applied Sciences, AIMS University. Our AIMS Biotechnology Students Association Advisor, Mr. Jivendran Sundarashekar, professors, lecturers, ladies and gentlemen, thank you for joining us. On behalf of the AIMS Biotechnology Students Association, I welcome you all. I'm very delighted to see all of you here, though it is virtually. This is our 9th webinar as part of our ABSA Webinar Series. As an association of students, ABSA always strives to provide a quality and comprehensive social platform to our biotechnology and bioinformatics students of AIMS University. We help them to learn new things, to grow, and having fun in the process. We have organized numerous activities, including the annual sports event, ABSA Olympics, faculty prom night, undergraduate orientation, to name a few. In this age of modern technology and the ever-growing field of biotechnology, our intellectual capacity is essential. However, our soft skills are equally important. In fact, soft skills are more important than ever before. No matter whether you are a researcher, entrepreneur, patent agent, or right there after graduation. Therefore, ABSA has started this webinar series. All students, especially the ABSA members, will have an opportunity to organize a webinar as a part of the ABSA webinar series. Our webinar series aims to provide a platform for the students to develop and refine their various soft skills. For instance, leadership, teamwork, communication, interpersonal skills, event organization, and time management skills. This webinar series are really student-centric and helpful for involved students in developing and sharpening their soft skills. At the same time, these webinars are helpful to ABSA members and all participants to update themselves with the latest knowledge in respective areas of webinar topics. I want to thank our honorable speaker, excuse me. I want to thank our honorable speaker, Associate Professor Dr. Rajesh Ramasamy. In fact, we are honored to host you. Thank you for accepting our invitation and becoming a part of our webinar series. Thank you, Professor N. Ravichandran, our Dean of Faculty of Applied Sciences, for the full support. I want to thank the organizing committees for doing a marvelous job. The efforts and time they committed to this webinar are appreciable. Last but not least, my special thanks to our ABSA advisor, Mr. Jivan, for taking this initiative to start this student-centric webinar series. Despite his busy schedule, he has trained and guided my team members by spending lots of time and energy. Thank you, Mr. Jivan. Ladies and gentlemen, once again, thank you for joining us. Without further ado, Mr. Tan Ai Kang, over to you. Please lead the way.
Thank you, Miss Vivian, for the wonderful welcoming remarks. Next, I would like to call upon the organizing committee chairman, Miss Vinodini Unaran, to say a few words and introduce the moderator of today's webinar. Miss Vinodini Unaran, the floor is yours.
Thank you, Mr. Tanaika. Greetings of the day to all of you. I'm Vinodini Kunalan, and I'm proud to tell you that I'm a second-year student of BSc Honors in Biotechnology degree program of AIMS University, Malaysia. It was an honor to serve as the chairperson of the organizing committee to organize this webinar. On behalf of the organizing committee, I welcome today's speaker, Associate Professor Dr. Rajesh Ramasamy, who is Head of Pathology Department, University Putra Malaysia, the Dean of our faculty, Senior Professor M. Ravichandran, the moderator, Ms. Sangeeta Balakrishnan, and all the participants of this webinar. This is the 9th webinar of our ABSA webinar series, organized by AIMS Biotechnology Student Association, and we are proud to host it. As a chairperson, I would like to express my sincere appreciation to our committed team who work hard to organize this webinar. Thank you, Associate Professor Dr. Rajesh Ramasamy, for accepting our invitation and becoming a part of our webinar series. Thanks to the Dean of our faculty, Senior Professor M. Ravichandran, for attending the webinar and supporting ABSA. I want to extend my thanks to Mr. Hugo, the President of ABSA, and Ms. Vivian, Vice President of ABSA, for facilitating in organizing this webinar. Last but not least, I want to especially thank our lecturer, Mr. Jivendran Sundrashekhar. Our committee members are lucky to have him as the advisor for ABSA. He has guided my team throughout the process for making this webinar a reality. I thank Mr. Jivendran for his guidance and molding us to play these roles. His determination, help, and see students excel is truly appreciated. Ladies and gentlemen, once again, I welcome you all and thank you for joining us. Mr. Tan Aikang, over to you.
Thank you, Ms. Vinodini Unaran. Now, I invite the moderator of the webinar, Ms. Chanita Balapinan, to introduce the speaker and start the session. Ms. Chanita Balapinan, please.
Thank you, MC Mr. Tan. Good morning, good afternoon, good evening, wherever you are. I hope you are having a good day. I am Sangeeta Balakrishnan and I'm proud to tell you that I'm a Year 1 BSc Honours in Biotechnology student at AIMS University. It is my honor to serve as moderator for this webinar. As moderator of this webinar, I also want to thank the audience for joining us. Just a gentle reminder, if you have any questions about the speaker, please post them in our Zoom chat box so that the speaker can answer them during our Q&A session. Every year, there are many people dying because of disease. I heard that there's this one particular technology that can save life against various diseases, which is like lymphoma, leukemia, to name a few. And that technology is the stem cell technology. Stem cell technology is a very powerful technology encompassing multidisciplinary techniques, and it is used in medical field to treat certain diseases like cancer. But how does it actually work? How stem cell help doctors in treating them in disease? In what types of disease can stem cell be used? Well, worry not. In today's webinar, we will have an excellent opportunity to get the answers to these questions and learn more about stem cells. We are lucky we have a prominent speaker from Malaysia. It is my great pleasure to introduce the speaker for today's webinar, Associate Professor Dr. Rajesh Ramasamy. Dr. Rajesh Ramasamy is an associate professor and head of department at Department of Pathology, University Putra Malaysia, UPM. He studied the degree in biomedical sciences at his University Kebangsaan Malaysia, UKM, and his PhD in stem cell transplant and immunology with a specialty in stem cell and immunology in London. For now, he is the head of Department of Pathology at University Putra Malaysia, UPM. With this brief introduction, I would like to invite Dr. Rajesh Ramasamy to take control of the floor. Dr. Rajesh Ramasamy, the virtual floor is yours.
Thank you so much, and I'm very happy to see all these young blood and also the young future scientists arranging such a wonderful talk on recent developments in the biotechnology and biomedical sciences. And also my sincere thank you to Prof. Ravindran, as a good old friend of mine. We used to sit for a meeting at the MOSTI, and also the MC, the chairperson, the coordinator, the many young blood, and which make me to feel young as well. And today I would like to talk on the new frontiers in the medicines, which is the stem cell, and later I will share my slide. And before to go to my talk, okay, so I would like to explain, or I would like to reinstate that this talk all based on my research. As you know, that stem cell being used in many medical fields. You say to any disease, people have tried to use the stem cell to cure or to leverage the disease level. But in this talk, I will be more focused on the research which I have done using a stem cell in a certain disease model. One is about how to modulate the immune response. The second one is on cardiovascular. The third one is diabetes. And the fourth one, I will tell what latest happened in the world that people have used stem cell to treat the COVID-19 disease when they would like to treat the lung failure. As you know that in COVID-19, that ultimatum will be the lung failure. So how we can use the stem cell to combat the immune response and also restore the lungs function. So my talk will be very technical, but I will try to use very simple word, not jargons, and I will try to be very casual. Don't think this like a lecture. Just think this like a discussions, and feel free, okay? Feel free to talk. Feel free to ask me if you think I speak too fast. Hold me for a while, and I will repeat. So don't take very seriously on the flow of the talk. Okay, if you don't understand, raise your hand or put you in the chat box. I'm much happy to be yourself. Okay, so I would like to share my screen now. Let's see. Can you see my slide?
Yes, Prof.
Oh, okay, that's good. That's good. So you can see my slide, right? Okay. Now let's talk about today's topic: a stem cell, a new frontier in modern medicine. Okay, as what your facilitator said just now, I think Sangeeta has said that many people die because of many diseases nowadays, and what the solution to uplift the human health in this modern world. And one of the solutions that been heavily investigated is the use of the field called regenerative medicine. When I say regenerative medicine, it is not new than stem cell. Okay, let's talk about regenerations. I'm not very sure how much of knowledge you have for the regeneration, but basically a regeneration is a process of production of new cell, tissue, and organ. And most of us, okay, most of us have seen these little organism in our house, okay, we call chicha or lizard, okay. So have you seen that? I mean, many people scared of lizards, right? Have you seen that in a very how they say, in a very critical situation, the lizard will chop, will what you call, will lift the tail of that lizard, and it will what you call, how do I say, camouflage? It will as a technique to what you call, to protect from that predators of the lizards, right? So have you ever think what happened in the lizard tail? Why lizard can dissociate the tail, and it can what you call use as a one of that technique to protect from that predators, and how could the lizard can grow back the tail very quickly? Okay, so these are things always amazing us, right? And also flatworms. Flatworms, the kind of that worm, when you chop them, when you split them into two, it can grow back into one entire organism. So do you ever think that if I can chop your hand, can you grow back your hand? If I chop back, if I cut off your leg, can your leg can grow back? See, these are things we lost during that evolution. So being the Homo sapiens, we lost many things. However, the lower category of the organism still keep the regeneration. So when you see a lizard can grow the tail, and flatworms can regenerate the entire new flatworm from the halfly split, this is called process regeneration. Regenerations, okay, happen in many organisms, but it depends on the level of evolutions. In the case of the human, regeneration happened, okay, in the liver most of the time, and also in the wound healing. So apart from that, there is not much of regeneration unless for the tissue level, where each tissue have their own stem cell. So due to wear and tear, the tissue and the organs in that particular organ, they can regenerate. And other example of regeneration will be the wound healing. And what are the magic bullet in the regenerative medicines? This called as a stem cell. Okay, stem cell can be found in any organs in a human body. And this we call as a tissue resident stem cell, and they able to differentiate into multiple type of the mature cell. Okay, and basically, just to recap, what is a regenerative medicine? Is a knowledge of using multiple approach to repair, to replace, and importantly to regenerate the impaired tissue or organ that resulting to restore and establish its normal function. That's entire process called as a regenerative medicine. So in the regenerative medicines, we have three components. One is tissue engineering, and biomaterials, and stem cell therapy. These are very focused on research, and today I will be more focused on how stem cell therapy can contribute to that, the what you call the high speed progress of regenerative medicines. And what is a stem cell? Now let's be back to a stem cell before we can talk about the usefulness of the stem cell for many disease model. Let's know what is a stem cell. Stem cell. I mean, all of us know that we have been created from sperm and egg. The sperm with the end nucleus and the egg from the ovum from the mother with the end nucleus. When both being fertilized, it forms zygote with the two end. and the two end will further differentiate into a multiple cell, and they form blastocyst. From the blastocyst, we will have the embryo, and embryo become fetus. It's a natural process. So basically, you and me are the results of the stem cell. You have to imagine, okay, in the initial day, you were as a one cell. So from one cell, how you differentiate into a billions and trillions of the cells, which has a different function, different morphology. How one cell become a liver cell, how it become a neurons, how it become cardiomyocyte. Do you ever think about that wonder of that natural, the wonder of the God that what you call create us from one single cell, which become trillion of cell with a different function, okay? That's due to a stem cell. Okay, so stem cell found even at the early phase of the human developments. Okay, if that sperm and fertilized sperm and egg being allowed to grow, it produces a fetus. However, in the laboratory, we also can produce that embryo, where the embryo can be used to produce, okay, not letting that embryo to develop into a fetus, but we can take that embryo to produce a specific cell that can cater to future medicine. Let's say someone is having a kidney failure, we can produce a kidney cell, nephrons, okay, kidney cell, or the kidney associated cell to form a kidney in the outside, okay, or can print the kidney to be transplanted into the patient. So these are actually in the research wise, okay. So now you got the point, what is stem cells, right? And if I say myself is a Rajesh, right? So it means I'm Rajesh, my father is Ramasamy. That's how I define myself. So how we define the stem cell? Not like any other cell, you do not need to define them because you have their origins and they grow. Let's say when you started to work with the cell line started to grow without any pot. They're going to grow continuously. However, this we call somatic cell. But stem cell, okay, when you say stem cell, the stem cell must have these two features, okay. One is they able to self-renew, and second thing is they able to differentiate into a specialized mature cell. I will explain to you later why they have to have these two features. Okay, remember, in your body, you have a stem cells, right? Let's, for example, for bone marrow. In the bone marrow, you have a multiple type of stem cell, and one of them is hematopoietic stem cell. Remember, we don't have too many stem cells. We have in our entire body, in each organ, we have a limited number, a limited pool of stem cell. But let's say our blood, right? Our blood will be replenished every 3 months, right? 3 or 4 months, every 3 months, right? I think every 4 months. Every 4 months, the blood of our body, the RBC will be diminished, and we will have a new blood, new RBC, right? So how the body will produce a new red blood cell every 4 months with a very limited number of the stem cell? Because in order to stem cell to become a mature cell to produce the red blood cell, that stem cell, or we can specifically say it as a hematopoietic stem cell, they have to propagate into a certain number. Okay, so they have to divide into a certain number. So some portion remain as a stem cell and some portion will differentiate into a RBC. So with this cell renewal means a process that make an exact copy of the stem cell. So before stem cell differentiate into a mature cell, the stem cell has to self-renew to produce a sufficient number of stem cell before to be used to mature into a mature cell. That's why cell renewal is very important. If you don't have a cell renewal, all the stem cell in your body will be exhausted. The end of the day, you don't have any stem cell, then you die because of degenerative disease. Okay, that's why cell renewal is very important. And stem cell can be retrieved from many organs and many sources. The first one is embryo, and we also can produce stem cell from fetal tissue, those fetal tissue which you can obtain from a legal, clinically aborted fetus. We can produce a stem cell, and most importantly, we also can produce a stem cell from perinatal tissue. Peri means during, natal means delivery. So during the delivery, what you get is either you will get your placenta, umbilical cord blood, or umbilical cord tissue. So these three things can also produce a stem cell. And then once the baby is born, that's we call post, postnatal life, whereby every organ in the body will have the stem cell. And our talk will be more focused on adult stem cell. And adult stem cells are multi, oligo, or unipotent stem cell. I will teach you later what is a multi and oligopotence. And the common adult stem cell are actually come from the bone marrow, which is hematopoietic stem cell that produce blood, and also mesenchymal stem cell that involve in providing a stromal support to the tissue and also immune modulation. So in the brain, we have the neural stem cell. In the liver, we have an oval stem cell. In the muscle, we have a satellite stem cell. So basically, every organ in our body has a specific stem cell or tissue resident stem cell that take care of that tear and wear of the organs, okay? And now, let's talk about how we can classify. So we know that stem cell can be retrieved from the embryo, fetus, and also cord blood, and also from the adult. So when we receive the stem cell from different kind of a life phase, they have a different character as well. And one of the characterization of the stem cell is the ability to differentiate. Not all of them, like your mother, right? Your mother, she's being mother to you. She's being a wife to your father. She's being a daughter to your grandfather. She's being a sister to her sister, a brother. So one single mother, but she has many multiple facets, right? Same thing as well, okay? So a stem cell may have a different potency. This potency we call totipotent, pluripotent, multipotent, and progenitor cells. Like a zygote, the first cell that derived from fertilization of the sperm and ovum, we call zygote. Zygote as a totipotent. Totipotent means you just split the word toti and potent. Potent means potency. Toti means total. It means when you take the zygote and when you implant into a uterus, that zygote can develop into an entire human being. That's we call totipotent stem cell, okay, or zygote. The next one is when the zygote develop into a blastocyst. In the blastocyst, you have an embryonic stem cell. This embryonic stem cell called as a pluripotent. It means they can become any cells in the body. However, when you implant these stem cells into a uterus, they cannot produce human being because they cannot produce the placental cell. They cannot produce extra embryonic layer, which is a placenta. Okay, the example of the totipotent stem cell are embryonic stem cell and also inducible pluripotent stem cell. Okay, these two are not so much important for us for today's talk, but what is important to us is a multipotent stem cell where you can get them from the fetal cell, perinatal stem cell, and postnatal stem cell. Okay, these are hematopoietic stem cell, mesenchymal stem cell, and any stem cell from the organ. So they can become a few type of the mature cell. It means like a pluripotent stem cell, embryonic stem cell, they can become brain cell, they can become a blood cell, they can become a muscle cell, they can become any cell of the body. But multipotent stem cell, they only have limitation for the differentiation. They only can differentiate along to their lineage. If the hematopoietic stem cell, the hematopoietic stem cell only can differentiate into a blood cell, like a red cell, white cell, and platelet. Like a mesenchymal stem cell, it only can differentiate into a mesodermal origin, which is a fat, chondrocyte, and osteocyte. Okay, so they have the limitations. Okay, like a cardiac stem cell, they only can produce a cell that in the heart tissue. Okay, and then we have progenitors. Progenitors are not a stem cell. These are and transiently existing cell in the system. So before stem cell become into a mature cell, the phase between the transformation, the translation of the stem cell into a mature cell involve the progenitor. So they basically will have a very limited differentiation ability. So I hope all of you understand what is the potency of a stem cell. So you should know that stem cell means it has two characteristics. One is they able to self-renew. The second one is they able to differentiate into a multiple mature cell type. And stem cell also have a potency based on where they derive from. It will have its own potency. If it is from that fertilization zygote, it is a totipotent embryonic stem cell, and inducible pluripotent stem cell have pluripotency, and most of the adult stem cell or postnatal stem cell, they have multipotency. Now, why stem cells suddenly become very what you call sexy, become very lucrative? Because two things happen in the entire world that change the perspective on the stem cell. The first one is Dolly. You know, Dolly is the first animal that been cloned. So the Dolly was born on 5th July 1996. It has three mothers. It means the Ian Milmuth and his colleague, they produced Dolly. So it's the first what you call cloning in animals, showing that three mother. Okay, it means three mother being used to produce a single child. It means ovum of one sheep are taken, and nucleus from the ovum of the another sheep are taken. These both are combined to make the zygote, and this has been implanted into a third sheep. So this is what we call cloning. It means that productions of the organism does not involve the involvements of the sperm and the ovum. It is just involved in a one somatic cell. It just involve the only single cell. Okay, that is we call cloning. And later in 2012, it's a very big boom, because usually what we know is when stem cell become mature cell, it's a one-way process. You cannot reverse the mature cell into a stem cell. It's always one-way process. And because of one-way process, we have a difficulty to acquire a stem cell for the treatment and research. But this wonderful man, Shinya Yamanaka from Kyoto, Japan, he shows that by using one somatic cell, the fibroblast, okay, he used the fibroblast, which is the most common somatic cell. By adding 4 different oncogenes, he convert the somatic fibroblast into a stem cell that has pluripotency. It means this pluripotent stem cell now can differentiate into any kind of a cell. So it gives a huge perspective that in future we can produce our organs or cell just can converting one of our fibroblasts into a IPS. It's all just a concept in the science and research. And this is just a simple explanation how embryonic stem cell being generated. So we have that fertilized egg with the sperm. It become a totipotent stem cell, okay, and it become an embryo. In the embryo, we have an inner cell mass. This inner cell mass, if you leave them, it become a fetus and human being. But if you take the inner cell mass, okay, if you disturb this embryo, you take the inner cell mass, and this become an embryonic stem cell. So this embryonic stem cell, okay, when cultured on the stromal cell or on the proper culture media, they can become any cell what you want. It can become a renal cell, it can become a liver cell or heart cell, whatsoever cell. Okay, and this is what we call the Sinmaya's work, showing that he produced that first what we call pluripotent stem cell from mouse embryonic and fibroblast culture. Okay, so what he has done is, okay, he This is what we call inducible pluripotent stem cell, or iPSC. Okay, why is this is so much of attract attention is it changed the concept of the stem cell. So basically, what we know is we can produce the stem cell will be what you call harvested from that in a body, and then it become a mature cell. So the process is a one way. It cannot, there is a no reverse, there is a no two way. But he, with an excellent and elegant experiment, okay, what he has shown is he had taken that adult fibroblast cell or fibroblast cells, and then he add these 4 oncogenes, okay. When you add this oncogene and he reprogrammed the fibroblast and it become an iPS cell, it means the normal cell, the somatic cell, acquire a feature of the embryonic stem cell. So this stem cell and now termed as inducible, because you induce a pluripotency by adding the 4 oncogenes into the fibroblast. And then this iPSC can be differentiated into any type of the cell, okay, by adding an appropriate induction media. Okay, so imagine that, okay, although this is not fully being translated into clinical, just imagine that you can take your fibroblast through your oral cavity, you add this 4 gene, and boom, it becomes a stem cell. So let's say if you're having a kidney failure, okay, you don't have a kidney. So either you will get the kidney from the donor. Now you have a chance to create your own kidney from your own somatic cell that been transformed into a stem cell. You just imagine, although this still like science fiction, many research are actually on the way to translate this into a clinical trials. And also, why iPSC is very important, because iPSC can be also used as a disease modeling. Like most of the time, we have some some rare genetic diseases or other genetic diseases where we can produce a specific patient-specific iPSC, and we differentiate them into a specific disease cell, and we can use our target's drug to do a drug modeling, and also we can use this to treating the patient. At the same time, let's say a patient having a degenerative diseases, you can edit, okay. Nowadays we have a lot of genome editing tools like CRISPR, and we have a lot of genetically modified or correcting method. So with that method, you can produce the iPSC, and then you can produce a particular cell that can be used to as a cell replacement therapy. So we can use iPSC for 2 circumstances. One, to do a drug developments, to a customized drug development to a particular patient. So nowadays it's a precise medicine whereby the medicine, the treatment are given to you based on your own cell, okay? Not like generalized, like you use Perinatal for everyone, but this is very specific, a tailored kind of the what you call treatment that given to you. Or we also can produce spare organs or spare tissues to replace the organ that damaged or the tissue that been injured or damaged. That's the beauty of iPSC. So I didn't go so detailed on this part because it just a theory, and it also shows that the prospect and the potential of the iPSC in the modern medicine. And also like in the cell-based therapy, like skin fibroblasts can be reprogrammed into iPSC, and that can be differentiated into thyroid follicles, and that can be used for cell therapy, disease modeling, or drug discovery. So you do not need to use a human or sometimes an animal for your research. So you can produce a specific cell that cater to the patient to have a precise result for precision medicine. And now, so we have a lot of ideas. So we have a lot of discovery that we have iPSC, we have embryonic stem cell, and we also have adult stem cell, okay? And then how this being translated, okay? The translation has been done after conducting many preclinical animal study and in vitro study, and then it has been translated into clinical trials. Okay, clinical trial has 4 phases. Okay, before a treatment become a treatment, it has to go through many phases of the clinical trials, at least 4 phases. Okay, so after the stem cell research being undergoing clinical therapy, it can become into a legalized therapy. Okay, so I have done the basic part of the stem cell. So I have explained to you what is a stem cell, how do you define stem cell, and also the prospect of the stem cell in the modern medicine by giving an example of iPSC. Okay, iPSC is still a bit controversy. Why? Because the embryonic stem cell and also iPSC, when implanted into an animal model, it produced teratoma, a cancer. Okay, due to safety issue, the usage of the embryonic stem cell and also iPSC is very limited, and they are still under that research. However, there is another category of stem cell which I mentioned earlier, which called as adult stem cell. They don't have this teratoma formation. They are adult stem cell, and their differentiation are very limited. They are multipotential. Because they're multipotential and these cells are actually harbored from that adult tissue, it doesn't have the character of forming cancer or tumor. So many people nowadays are utilizing adult stem cell for their research and many clinical trials. If you check into the FDA and also the American Clinical Trial Portal, you can see the many adult stem cells being investigated for the multiple therapeutical purposes in the modern disease. Okay, and now this is my main work. This is what my PhD. During my PhD, I have investigated the impact of the stem cell in modulating the immune system. Okay, when you say immune system, so we have hematopoietic stem cell. That hematopoietic stem cell can give rise to many mature immune cell. It can be T cell, B cell, NK cell, neutrophil. It can be also the eosinophil and basophil. So we just put away the red cell and the platelets. So we will be more focused on the white cell. And why we need suppress the immune response. Okay, you have to know that not all immune response are favorable. Sometimes we have the autoimmune diseases whereby your immune system recognize your own body as a what you call as a foreigner, as an enemy. So it try to attack your body. So it will create the autoimmune disease. Okay, the autoimmune disease also can be the type 1 diabetes, and we have multiple sclerosis. Okay, and we also have other autoimmune diseases. Okay, so we can what you call say it continuously. So these are autoimmune diseases whereby the immune system recognize yourself as an enemy, and sometimes you also have to control the immune system in the case of transplantation. The transplantation, basically, the individual, the receiver will receive an organ from donor. However, the immune system of receiver, okay, you may can what you call fool the police, and you may can fool this the spy, but you cannot fool the immune system. Your immune system has high intellectual property. It can recognize any single foreign molecules, any single foreign cells in your body, and it will work so hard to eliminate that foreign cell. So that's why in the case of transplantation, when you don't have a fully compatible matching, when you have average matching, there is a higher chances that the immune system will reject the organ that been transplanted. And sometimes you have a chronic immunosuppression, which is imposed risk for infection. And how we can see the reason how we can evade the immune system. So we can give the patient into chronic immunosuppression by steroid drugs. But these steroid drugs, they have their own side effect, and it's also imposed for a high infection, high risk for infection. And what will be that alternate way way to what you call induce immunosuppression when needed. So that's how it's come mesenchymal stem cell. Mesenchymal stem cell is originally found in the bone marrow. Now you can find the mesenchymal stem cell any organs of your body, and these stem cells deliver a non-specific immunosuppression for a short time period. Okay, so you do not need to have immunosuppression for a longer time, you just need them for transiently. Okay, now the mesenchymal, as I mentioned earlier, all the stem cells, okay, need to be defined. So how we define mesenchymal stem cell? Mesenchymal stem cells should be positive for some markers like a CD105, CD90, and is also negative for the other markers like a CD45 and 34. They differentiate into a multiple cell type, and they also express the markers that indicate that they can differentiate into a multiple cell type. Okay, and so here what we have done is we investigated, okay, the effect of the mesenchymal stem cell on the lymphocyte. Okay, so my PhD is to control GVHD. GVHD means graft versus host diseases. So when we do a stem cell transplantation, what happens is the stem cell differentiate into T cells and B cell, and these T cell and B cell actually came from donor. Now the donor or the graft imposed an immune attack on the host cell. So how to control this GVHD? So this can be controlled by using mesenchymal stem cell. So what we have done is we co-culture the lymphocyte, okay, from the donor, from the graft, and with the mesenchymal stem cell we co-culture them. Okay, after we co-culture them, and we measure the proliferation of the lymphocyte. So when we activate the T cell with the stimulator CD328, okay, the T cell proliferate very vigorously. But when we add more stem cell, the proliferation reduces. When the concentration or the number of stem cells that been added into the co-culture reduced, the inhibition also reduced. Okay, so it shows that the mesenchymal stem cell can suppress the proliferation or immune response of the T cell. So this immunosuppression it is not confined to a particular type of T cell. Both CD4 cell and CD8 cell are equally been affected. Okay, this shows that the purple color one T cell with the stimulation, and the red color one is T cell with the stimulation with the mesenchymal stem cell. So without any suppression, see, the T cell can proliferate how many rounds? 1 round, 2nd round, 3rd round, 4th round, 5th round, 6th round. So they can proliferate 6 times. But when we add MSC, you see the number of the proliferation and also the volume of proliferation reduced in the presence of the MSC. And it is applicable for both CD4 cell and CD8 cell. Okay, so it shows that mesenchymal stem cell, regardless the type of the T cell, and also any T cell or B cell, NK cell, or neutrophils or monocyte, macrophage, DC, they able to suppress the immune response. So our group, the stem cell and immunity in UPM, we are pioneer in this research. We have conducted the impact of the MSC on that many type, an array of the immune cell, and we found that mechanism and how the MSC actually reduced the immune response by targeting the proliferation, activation, and effective function of the immune cell. So that's the first part of my my talk on immunomodulation is completed. Now we move to a second disease model. So we also work with the diabetes. As you know that diabetes can be classified into a two type. One is it all depends on two things, and the quality and the quantity of that insulin. When the quantity of the insulin is reduced, when the body could not produce sufficient amount of the insulin, you will have a type 1. when the quality of the insulin is reduced, whereby you have sufficient amount of the insulin, but the insulins cannot act on that target cell. Okay, they're not sensitive to the insulin receptor, or the quality of that quality we call type 2 diabetes. Okay, so the diabetes and one of the NCD, non-communicable diseases, which affect a huge population in this world, and so much of the research work and clinical trials are going on to cure diabetes. However, until now there is no cure to diabetes. We only can manage the diabetes, and the way to to treat the diabetes is either you restore the pancreatic beta cell, or you can control the autoimmune attack by the autoimmune immune cell. But restoring the beta cell is quite a difficult process, whereby you need a cadaveric islet transplantation, but it is not an easy process. Okay, and also it's a painful process, and sometimes you will never find a fully matched pancreas. Okay, and also if it is a transplantation involved two different genetically individual people, then you require continuous immunosuppression. And sometimes, okay, beside having an option to the cadaveric islet transplantation, it's also possible that to produce a beta cell from stem cell. So many people have said that the pluripotent stem cell, for example embryonic stem cell and ips, can produce a beta cell, and also people have used the mesenchymal stem cell to reduce the inflammation. Remember, the type 1 diabetes and also sometimes type 2 diabetes is also due to the chronic immune response. So by using the mesenchymal stem cell, we can ameliorate the immune response and restore the pancreas function. So many people have, there are certain clinical trials being conducted, and it shows that MSC can be safe to be used. Either they come from same patients or from the different individual, and both MSC, either autologous and allogenic, are safe to the patient to be transplanted. And there is so many of clinical trials are actually being conducted, and the results are actually very varied. Okay, the results from these clinical trials show very mixed result. But to conclude what we have found using the MSC in that diabetic treatment. So what they have shown is MSC doesn't differentiate into a beta cell. Okay, they never find that MSC can differentiate into beta cell, but what they have found is MSC reduce the inflammation in the pancreas. When the toxic environment such as inflammation is produced in the pancreas, the tissue resident stem cell within the pancreas, they can revive and they can give rise to beta cell. And second thing is MSC also can recruit and revive that tissue residence beta stem cell. Okay, and that's one thing. And second thing also, people also using new way of treating the MSC. These are still under clinical trials, whereby the patient's T cell and B cell that is reactive towards the pancreatic beta cell are being educated by the MSC. So now they're not being reactive towards this beta cell. And then my third one is a myocardial infarction and heart failure. And about 17 million people die every year. And it's one of the most common causes is ischemic heart disease, is imbalance of the oxygen supply and the demand. It kills, so when there is a myocardial infarction, it kills 25 of the cardiomyocytes, okay? So usually, so when you have the death of the cardiomyocyte, you have a two option, okay? One is a quick fibrosis, other one's a regeneration. As you know that heart is post-mitotic organ, it means that it don't have so much of regenerative capacity. So in this case, what happens is the heart will take a quick what you call way to do a fibrosis. But fibrosis is not a cardiomyocyte. The fibrocyte cannot what you call contract. Okay, so in order to have and a fully functioning or optimally functioning heart, you need to have a cardiomyocyte, not the fibroblast. Fibroblasts serve as a quick fix for the injury, but it cannot function as a cardiomyocyte. That's why in the case of the myocardial infarction, where you have a fibrosis, and most of the patient will have lowered cardiac function. And the moments we have a pharmacological therapy and also revascularizations. Okay. However, this cardiac regeneration can be achieved by using multiple type of the stem cell. Okay, we can use a scuttle myoblast or bone marrow stem cell. Okay, it can be any bone marrow stem cell. It could be endothelial cell. It could be a hematopoietic stem cell, or we can use a mesenchymal stem cell, or we can use the induced pluripotent stem cell, or we also can retrieve the tissue resident stem cell of the cardiac tissue, or it can be also embryonic stem cell. So people have used multiple type of the stem cells to regenerate the infarcted area. Okay. And this is one of our research we have conducted many years ago. So we receive, we have conducted this research work where for the last stage of the cardiac patient. So during the open heart surgery, we receive a small piece. Okay, it's very small piece of the cardiac tissue. From the cardiac tissue, we generate that the cardiac stem cell. So by day 10, we can achieve a sufficient number of the cardiac stem cells, and we characterize the cardiac stem cell using specific antibody like a STRO1 and CD29 and also CD105. And we also look at that expression of the stem cell markers and also expression of the cardiac lineage marker. So we are successfully had generated the cardiac stem cell from that a small piece of tissue of the cardiac tissue that came from the patient. And then what we happen is, so then we produce them for within a month time about 1 million of the stem cells, and then it be potentially to be used in the clinical trial. So it shows that for the cardiac defect and cardiac related degenerative diseases, okay, in order to avoid the fibrosis, okay, fibrosis is a quick fix for the myocardial infarction, but the fibrosis cannot restore the function of that cardiomyocyte. That's why we need to replenish that infarcted area with that, either by the cardiac stem cell or cardiac stem cell derived cardiomyocyte, or using any other stem cell. So this will restore the function of the cardiac cell. Okay. Now the last part of my talk will be how the mesenchymal stem cell being proposed to treat the modern disease which we have now, which is a COVID-19. As you know that SARS-CoV-2 is a novel coronavirus induced pneumonia, which named as a COVID-19. Okay. This virus, okay, has as an envelope virus with a spike protein, a single-strand DNA. The envelope has a spike glycoproteins bind to the cellular receptor angiotensin 1 converting enzyme to ACE2. Okay, you can see very clear. And most of that the vaccine nowadays are being produced against these spike proteins. So what happened is, usually a normal person, a person with that optimal immune response, the body can withstand the disease, and the body can heal, can produce an antivirus response in order to avoid that advanced stage of disease. Let's say patient with immune compromise, patient with a comorbidity, older patient, young patient, where they don't have a fully functioning immune system, it can cause them advanced level of pneumonia. It means it may cause lung failure. Okay, so in the case, what happened is the body start to produce multiple pro-inflammatory cytokines, and these cytokines produce a cytokine storm that cause a collapse of the lungs or the alveolar cell or the epithelial cell. So it means the lung will lose the function, so it is unable to exchange the gas that require for the body metabolism. So in this case, what happened is, can we use, because in this case you have to remember two things has happened. One is the extra-generated immune response, the body fully affected by the cytokine storm, the pro-inflammatory condition. The second one is the injury of the lungs tissue, epithelial tissue. Can we address these two by using the mesenchymal stem cell? So when the immune system is overactivated, production of a large number of inflammatory factors resulting in a severe cytokine storm. And it induces organ damage followed by acute respiratory distress syndrome or ARDS or acute cardiac injury, and also which may cause death. So preventing and reversing cytokine storm may be the key to save the patient with a severe COVID-19 pneumonia. Okay, and avoiding the cytokine storm may be a key for treatment for COVID-19, and MSC, owing to their powerful immunomodulatory or immunosuppressive ability, may be beneficial to this attenuating the cytokine storm. So this is the first clinical trial being conducted in Wuhan, China. So they have used mesenchymal stem cell, okay, and before that, so what they have, because usually in that ARDS or the lung failure or the lung infection, what you will see is, you will see this kind of the ground glass opacity. So it shows that damage has been made, okay, as damage has been made in the patients. However, when they have used mesenchymal stem cell, see the damage is very severe. However, when mesenchymal stem cell to given to the patient, you could see that the damage made by the disease are actually disappeared. So it shows that either the MSC helped to restore the functions and also regeneration of the epithelial cell in the lungs, either through the process called restoration of the epithelial cell, or they also reduce the cytokine storm by inhibiting the immune response that due to COVID-19. Okay, so you can get, because I can go more detail, but I'm afraid that you may don't aware of the terminology. So I just summarize that the results which obtained from the first clinical trials in the China. And so there are many approaches of using MSC. Okay, one is cellular therapy, whereby we can use bone marrow, mesenchymal stem cell, umbilical cord stem cell, adipose tissue stem cell to treat that COVID-19. And sometimes we also can opt to the acellular therapy. It means MSC, because if you notice that the major therapeutical effect of the MSC actually come from the excretion, the soluble factor produced or secreted by the MSC. So what people have done is, people have cultured the MSC, people have their exosome. Exosome is a kind of a small organelles in the body that produced by the MSC. I mean, most of the cell produce the exosome. This exosome being excreted in that what you call culture media, and is being collected and given to the patient. Okay, so rather to use a cell to treat the patients, now people have moved to, they have taken that factor produced by the MSC to treat them. So this we call acellular. So we don't use cell, but we use the organelles, the cytokines, the growth factors produced by the particular stem cell to treat a particular disease. Okay. So why MSC lucrative for COVID-19? Because as you know, MSC, they can home to the lung and save. It is safe, and it can mediate the lung reparative function in animal model of lung injury. And they also can be immunosuppressive. Okay, they can suppress the entire immune system, and they secrete anti-inflammatory cytokine. They also show they produce antimicrobial substance towards the bacteria and viruses. Okay, and it's also inhibit the lung fibrosis. It regenerates the lung tissue, rescue the injured cell from the apoptosis, and enhance clearance of the alveolar fluid. And it's also produce extracellular vesicle that can be used as a what we call ACE therapy. However, there is a lack of evidence whether MSC can directly transform or transdifferentiate into epithelial cell is still lacking. So with that, I think I can end my talk. So what I have captured at the moment is that stem cell, okay, it has a huge prospect for treating modern diseases, chronic diseases, especially cardiac, diabetes, and even modulation. And many research are still under the what we call clinical investigation. So what I show is the prospect of stem cell. So when you listen to this, you may not surprise, you may what you call fully overwhelmed that stem cell can be done so much. I think yes, it can do so much thing. However, all this work should be properly subject to the clinical trials before it becomes a clinical application. So what I have explained to you, what I have mentioned to you just now, these are mostly are under the research or under the investigation, and where this has to undergo a proper clinical trials to become a clinical therapy. With that, I end my talk. Now I would like to have any questions from the, you may ask me the question from the floor.
Thank you, Dr. Rajesh Ramasamy, for the wonderful talk. Now we will have our Q&A session to answer participants' questions. I would like to invite the audience to ask questions to clear your doubts. Questions can be asked either by submitting it through Zoom chat box or unmuting yourself.
I think it's best to ask me directly. It's much interactive, you know.
Yeah.
Be brave, bold. You guys young, right?
Thank you, Dr. Rajesh.
Thank
for
you.
supporting our students. Nice to see you after a long time. It's totally run by students. So yeah, very enthusiastic and very nice. Nice to see.
As I told you, there are a lot of aspects. People have used stem cell for neuron diseases, skin diseases, but I only touch those which I work with that, okay? So don't think that the stem cell can be used for these 3 different kind of disease. It actually is huge. If you Google, the Google Auntie will tell you a lot of story than mine. Any part, maybe you may have difficult to understand certain terminology, you don't understand certain explanation. If you have that, you always feel free to ask me. Or if you, or maybe in the future you want to do a master or PhD in a stem cell, always feel free to approach me. Who knows? You could be my successor.
There has been few questions asked through the registration form. So the first one would be stem cell. They asked if the stem cell got any bad effect to humans or not.
Bad effect in the sense that what kind of a stem cell? You can't simply blankly or blanketly say a stem cell. Stem cell, we have embryonic stem cell and also inducible pluripotent stem cell, adult stem cell. See, like when you utilizing the embryonic stem cell and also inducible pluripotent stem cell, these are very volatile stem cell. It means these 2 stem cells can become whatever they want. So when you're young, okay, they're very volatile. It means it can be whatever you want, but same time, because of this very volatile, it also have a plenty of what we call system that control their differentiation. So many people, when these stem cells being transplanted into to measure the safety, they found that these stem cells can produce tumor. So that's why the usage of embryonic stem cell and also the inducible pluripotent stem cell in the clinical application and also in the research in a clinical trials are very less because safety issue. Although it has so much of the therapeutical prospect, but when safety is compromised, we can't use them for the research or even in that clinical application. Okay. And that's one of that, I would say that drawback of the stem cell, and especially those pluripotent stem cells. It's like, see, it's very simple. When you're young, when you're preschool, when you've been asked what you want to be, you'll say, I want to be a pilot, doctor, this and that, right? You're pluripotent. So let's say when you move slowly to your SPM, after going through all the trouble in life, and you know where you are. So at that time, you know, most probably I'm not into a pilot. Maybe I want to be a biotechnician. So you become a multipotent stem cell. Now, from the pluripotent to become whatever you want, you slowly evolve into one single ambition and ambition or what you want to be. That's what happened to the stem cell as well.
All right.
Because when you're young, you think you can change the world, right? You can change, you can do whatever you want. When you're 5 years old, even you believe in unicorn and Godzilla and dragon, right? And this age, you know the reality. You know there is no unicorn. You know there is no dragon, right? So you know you only have an iguana or lizard, something like that. So that's the only drawback with the stem cell, those with the pluripotent stem cell. And remember, in Malaysia, not only the hematopoietic stem cell transplantation for leukemia patient is illegal. Okay, other stem cells are not legalized. Okay, because in private, I have no idea how they actually doing. But in Malaysia, if you wanted to have a stem cell therapy, that therapy should have completed the clinical trials and also FDA approval. So remember that. So many work in Malaysia on stem cell are still under the investigation, either in a laboratory investigation or preclinical or under the clinical trials.
Also, sir, there are several supplements of these stem cells nowadays. So is it advisable to consume them?
What is that?
There are few supplements for stem cells.
See, you cannot consume stem cell. Whatever you consume, you'll be digested, right? What they meant is a stem cell enhancer, like a green algae, okay? See, the stem cell enhancer is not something new for us. In the Indian and Ayurvedic and Siddha medicines, we have the Gayakalpa herbs. That's a stem cell enhancer. See, in an Indian and also in Chinese medicine, they do have certain like a ginseng and certain herbs. They actually can enhance the number of stem cells in your body. So what these modern people are actually doing is they are preparing these components, a compound. It could be from any herbal formulation or mineral formulation. When you consume, it will increase the number of stem cells, but whether they are clinically proven, not sure, right? It's only just by being word of mouth. Maybe they have some little research, but it's not actual clinical trial or clinical investigation. But remember, our ancestors have done this. So in Ayurvedic and also in Siddha medicines, we call Kaya Kalpam. Kaya Kalpam means rejuvenation. Okay, and also certain herbs. I didn't include these in this talk. I do work on herbs and also stem cell, but I didn't include in this talk. But in our ancient system, we have our what we call specific nomenclature for the herbs that indicate which herb has this what we call rejuvenation ability. Same thing goes to Chinese medicine as well. Like an amla is one of the amla and turmerics are known to have what they call have regenerative potentials. And also maybe in the Indian like Karisalangani, which been used for that hair, it known to induce hair follicle stem cell proliferation. So yeah, can, but it not been fully scientifically proved.
And the next questions was, what are the obstacles that must be overcome before the potential uses of this stem cell in cell therapy will be realized?
Yeah, as I explained to you, you need a proper scientific investigation and clinical trials. Because clinical trial is not an easy process, okay? You require millions of money. right? Because you have to recruit the patient and patient being monitored, and it has been done with the different phases, phase 1, phase 2, phase 3, phase 4. And because of that, sometimes when we do clinical trials, whatever impact which you see in the animal is not being mimicked in the animal study. There are many what you call stem cell clinical trial being suspended because they don't see an expected impact on that human. So these are things, the lack of scientific study, the lack of clinical trials that hinder the stem cell therapy to stem cell treatment to reach the patients. And also it's not a very cheap treatment, because you cannot simply produce a stem cell in that lab. You require GMP lab. GMP lab itself require millions of dollars to have one, and the entire process. So maybe one vial of that stem cell, if let's say if it has been approved, it may cost from 10,000 to 30,000 of Malaysian ringgit.
There's a question in chat box. So the question is, stem cell use in surgery is rather aggressive in all fields of medicine or surgery. Do we have data to show their success rates, various adverse outcomes?
I mean, what is stem cell use in surgery? You need to be very specific. I mean, you mean the organ transplantation or the hematopoietic or that the bone marrow transplantation? See, like okay, let's I explain to you the one of the most successful stem cell therapy. About many years ago, when people have leukemia, what do you see in Tamil movie or in any Asian movie? Most of that, what you call in the climax, every hero or heroine will die of leukemia, right? Because they say there is no treatment. Okay, that's the old story. But you see, the treatment for leukemia has become revisited after the stem cell therapy. So what happened is nowadays we can do the hematopoietic stem cell therapy, whereby we can remove all these cancerous stem cell and also cancerous cell from the bone marrow of the patient, and we introduce a new stem cell from patients, okay, which free from cancer, and now the patient can have the donor's red blood cell and the donor's blood cell which is free from the cancer. So the hematopoietic transplantation for leukemia shows is a only treatment that can cure leukemia. So in that case, yes, it's work fine for that hematopoietic stem cell transplantation. But for others, not very sure, because for except for that hematopoietic stem cell transplantation, there is no other actual legalized stem cell therapy in Malaysia.
The next question is that, is there a situation known as chimera where a person is found that he or she has different type of cell?
Chimera, when you have a transplantation, let's say in the case of the hematopoietic stem cell, yes, your chimera. It means your blood cell actually come from the donor, the second party. But the chimera doesn't mean that you and the donor are completely different. See, when we do transplantation, we do tissue matching, right? You cannot simply transplant any tissue into an individual. Okay, most of the time we will get a closest match from the family members, the sibling. The most of the time, the chances to have closest match come from siblings or that haploidentical from the parents. So the chimera which you're going to see is very little. The differences of genetics is very little. Yes, it is. But not like something you having human head and lion body like what you see in the mythos. That's a different one.
I think that's all. about the questions?
No idea. If you have any questions, you always can email me. I may write my email address here, so you can email me as well. I'm much happy to guide or help. So I have, I have, wait, let me do, I have written my email address to all of you. So I just want to ask you, do you really understand my talk? Is it too technical? Be honest. Sankita.
Yes, to be honest, I feel like you summarized everything so that we students can understand that in a better way, like it's not complicated.
Because this is usually for those for the knowledge audience, but I have no idea how much you have learned in your course. So some of you from first year, but doesn't matter. You see, if you listen to a talk and you understand everything, it's no fun. If you know everything, why you should be in university? You should be working, right?
Yes.
Your students, you meant to be not know everything. So it's okay. Learn. Any questions?
I would like to know if Professor Ravi has anything to say about this. So I think that's all. So thank you, Dr. Rajesh Ramasamy, for answering the participants' questions. It was a great session. Before I pass the mic back to the MC, I would like to take this opportunity to thank the organizing committee for giving me this chance to serve as moderator. This is my first time in life I served as moderator. This experience helped me a lot to learn a lot. I enjoyed the role of being this moderator today. I sincerely want to thank our senior lecturer, Mr. Jivendran Chandrasekhar, who's also our ABSA advisor. Thank you, Mr. Jivendran Chandrasekhar, for our continued support. Ladies and gentlemen, once again, thank you everyone. MC Mr. Dan, over to you.
Thank you very much, Dr. Rajesh Ramaswamy, for a wonderful and informative talk. And thank you very much, Ms. Anita Malapitian, for moderating the session. Now, I would like to invite the Dean of the Faculty of Applied Science, Senior Moderator and Ravi Agent to deliver his opening remarks. Now, I would like to invite Mr. Lim Yenda to deliver a word of thanks. Mr. Lim, please.
Hello, everyone. I'm Lim Jenda. I'm proud to tell you that I'm a Year 3 student of BSc Honours in the Biotechnology degree program of Ames University. The organizing committee has given me this wonderful opportunity. First of all, on behalf of the ABSA and Webinar Organizing Committee, I want to thank Associate Professor Dr. Rajesh Ramasamy for an enlightening and informative talk on stem cells in modern medicine. Dr. Rajesh Ramasamy, we are grateful for the time and effort you took to share your thoughts and experience with us. We appreciate it. Thank you to Senior Professor Manikam Ravichandran, Dean of Faculty of Applied Sciences, for showing your support to us by attending today's webinar. I want to thank Mr. Hugo, President of ABSA, and Ms. Vivian, Vice President of ABSA, for their help. I would also like to thank all the participants from various countries for joining us for this webinar. I believe that this webinar helped you to get some insights and learn something new about stem cells in modern medicine. I would like to take this opportunity to record my thanks to all members of the webinar organizing committee for their hard efforts. Working together as a team for this webinar was an engaging and enjoyable experience. Last but certainly not least, my special thanks to our ABSA advisor, Senior Lecturer Mr. Jivendran Sudarsekar, for his valuable support. Ladies and gentlemen, once again, thank you. Mr. Tan, over to you.
Thank you, Mr. Lim. It was a great session today. Before ending the session, we would like to hear the feedback from all participants by sending the call on the screen right now. Report me, the evaluation survey will not take you more than 2 minutes. Your feedback will be greatly appreciated. You can either send the QR code or go to the link shown below. The link is posted in the chat box as well. After the completion of the survey, you will receive an instant participation certificate in a short while. And I will keep this slide running for 2 more minutes for you to send the QR code. We are looking forward to having you all join us again in the next session. Thank you again, everyone, and we wish you good health and safety.
Thank you.
Before we all leave, I would like to take a group virtual photo. So here we go. 3, 2, and
No, we just wait for 2 minutes for the participant to send the QR code.
all right.
hope everyone has the chance to send the QR code. And I'm going to end the meeting. See you soon in our next webinar. Stay safe and have a wonderful day. Bye for now.
Thank you. All the best.
Thank you so much, Prof. Thank you.
Thank you, Prof.
Okay, good job, guys. At least it's settled.
Thank you, thank you.
Bye-bye.
Bye bye.